in all directions, sir. Good afternoon, all. It's Gautam, and we are taking botany now, and we are continuing with this tomorrow. Means the same session what we have discussed in last class, that is sexual reproduction in flowering plants. And regarding this, we completed 2 sessions already. In initial session, we have discussed about the introduction of the unit and the introduction of the scientist. And in the next session, we have discussed how the flower is formed, and the androecium, that is stamen structure, we have discussed. And if you cut the stamen, the TS of the stamen or TS of the anther or TS of the microsporangium, how it looks and what it is composed of, that we have discussed in the last session. So as we know that we are discussing with this sexual reproduction in flowering plants chapter, and these are the analysis of the last year paper. So we completed this stamen structure also. We completed this TS of sporangium. And once again I will recall that, and no need to worry here. See here, this is the anther. When you cut the anther, it looks like this structure. And here, so this is, it is having 2 lobes. This is 1 lobe, this is another lobe. And each lobe is having 2 thecas. So this is 1 lobe, it is having 2 thecas. This is another lobe, it is having 2 thecas. So totally 4 thecas will be there. So as it contains sporogenous tissue here, this we call this structure as a sporangium. And this sporangium has 4 wall layers next to that sporangium. So you can see here the dense structure, the layer of this dense structure, what you are seeing, that is we call this as a tapetal layer or tapetum cells. The innermost layer of this microsporangium wall, it is tapetum cells. So these are tapetal cells or tapetum. So this special thing about this tapetums, so even in last year I displayed the last year question also in last session. In the end of the session, we discussed the PYQ, previous year question of this one regarding on this tapetum topics. So they asked on this, so 2025 PYQ it was there, the function of tapetum and the feature of tapetum cells there. So these tapetum cells, how do they look? So they are having dense cytoplasm. So they are having dense cytoplasm or protoplasm, and they are having 2 nuclei, so which is binucleate. So that is binucleate in stage, and abundant food material. Abundant food is there, which nourishes the developing pollen grains. So this is the sporogenous tissue, no? So what is this? The inside of this, what you are seeing, this whole content, what you are seeing, this is sporogenous tissue. This sporogenous tissue itself is going to become pollen grains later. So to develop from sporogenous cells into pollen grains, so that nourishment, it will be provided by these tapetal cells. So they will provide nourishment to the developing pollen grains there. So to provide the pollen grains, it has to secrete more amount of food in it, and it is having 2 nucleus there. So that is the special thing about the tapetal cells there. So next to that tapetal cells, there is an layer, there will be multiple layers will be there, that is we call this as middle layers. These are middle layers, and next to that middle layers, what you are seeing, the next to that middle layers, this we call this as endothecium. Endothecium, and the outermost layer is there, no, that is epidermis. So here, except tapetum, even tapetum also protects, but these 3 cells, they will be working together to it is also acting as protective unit, or for protection it will be protection of the developing pollen grains. Along with this protection of the developing pollen grains, it also helps in dehiscence of anther. Dehiscence of anther in the sense, when the anther matures, it bursts. It will burst like this. When it bursts, the pollen grains which are present in that, it will disperse. It means here it helps in dehiscence of anther. Dehiscence in the sense which will be losing the moisture and getting drying out. And especially when you see that case, in case of endothecium, these cells are hygroscopic in nature. Hygroscopic in nature. Hygroscopic in the sense which helps in drying out or which helps in losing the moisture. That is hygroscopic. Endothecium are hygroscopic in nature. And next to that, what you are seeing, there are 4 different microsporangium are there. And if you see the arrangement, how the arrangement is, this is polygonal structure. This is polygonal structure. And 4 sporangia are there. This is first sporangia, this is second sporangia, and this is third sporangia, and this is fourth sporangia. So togetherly 1, 2, 3, 4. 4 sporangia means tetrasporangiate. The TS of anther, it will be tetrasporangiate. And how the microsporangium looks like? This microsporangium looks like how it is looking. It is like circular in outline. So the microsporangium, it will be having circular in outline. So where you will get a questions. These are NCERT lines only which I am saying it now. So the anther is bilobed and each lobe is containing 2 thecas, and thecas also can call, we can call this as a sacs also. Theca is nothing but a bag or a sac where within that the sporogenous cells will be there. These are sporogenous cells which are present in that. Here sporogenous cells are there. Here sporogenous cells are there. Means these many thousands of pollen grains are produced from each sac there. So means thousands of pollen grains are produced when one anther bursts. So this is about TS of microsporangium or TS of young anther. So this sporogenous tissue is there, no? So this inside of this, what you are seeing, this is sporogenous tissue. So this is sporogenous tissue here. So this sporogenous tissue, I said in the last class. So this sporogenous tissue is going to become pollen grains. So this sporogenous tissue, it is going to become pollen grain there. Means after maturation, it is going to become a pollen grain. So here, this process, because of this sporogenous cells which is getting converted into pollen grains, then I will be calling this as pollen sacs. Pollen sac in the sense pollen bag. I will be calling it as pollen bag at the time. So this is about pollen grains and the sacs which are present in the microsporangium or anther there. So next we are going to discuss microsporogenesis. So as the name indicates, so understand the names there, wordings. Microsporogenesis. Here genesis in the sense formation. So microsporogenesis in the sense the process of formation of microspores. See, microspore word is there here. Microspore genesis in the sense forming. The process of formation of microspores, we call this as microsporogenesis. The process of formation of microspores, we call this as microsporogenesis. From where it is forming? From how it is forming? From microsporo mother cell. From microspore mother cell, or MMC we call this as. See, there is a microspore. I don't know who the mother is, but I will say no. If I see that, if someone is calling this, if microspore is calling that mother, then I will be calling this as microspore mother cell. So the mother of microspore is microspore mother cell. So here what is the definition says? Microsporogenesis in the sense the process of formation of microspores from microspore mother cell. When the microspore mother cell undergoes meiotic division, so that continuation through meiotic division, we call this as microsporogenesis. So are you able to understand what I am telling? So there is, and before understanding these lines, I will explain in simple way, you can understand there. See, in last session, in this slide, we have seen it is a sporogenous cell or sporogenous tissue is there. So the same thing I am writing here, sporogenous cells. Sporogenous cells are there. So these sporogenous cells, these cells which are 2n in structure, 2n in the sense of diploid. If you take our body cells, how the body cells will be? It will be in diploid content. 46 chromosomes will be there in each my part of our body, except gamete cells like sperms, eggs. So there the content of cell, the DNA content or the chromosome number, it will be in a haploid content. The cells except gamete cells, other than the gamete cells, all the cell composition it will be in diploid content. So now it is sporogenous cells which are present in this. So see, it is a TS of anther, and within the anther what you are seeing, these are microsporangium. So within the sporangium what you are seeing, these are sporogenous cells. So what it is? It is a young anther. So it is young anther. So now later it will become matured anther. Wait. So later it will become matured anther there. So later it will become matured anther. So initially it was only sporogenous tissue or sporogenous cells, means it is like all cells which is having diploid in content. This sporogenous cell, it will become microspore mother cell, MMC, which is 2n in structure. Means the same cell, it is going to act as microspore mother cell. Now it become a mother, means it increases in size of a cell. Initially sporogenous cell was like this. Microspore mother cell, it will become like this. Because it is getting prepared for what? It is getting prepared for cell division. We have seen in cell cycle and cell division in 11th class chapter, cell cycle and cell division. If you go and check there, you will get to know that nucleocytoplasmic ratio, it disturbs when the cell is undergoing division. So here, if the cell is undergoing division, means the cytoplasmic ratio, it increases. This is microspore mother cell. So this microspore mother cell, it will undergo meiosis or meiotic division. So it will show meiotic division. So it will show meiotic division there. So when it shows meiotic division, these microspore mother cells also, they know. So one cell is there. If it is undergoing meiosis, how many cells we will get? So if one mother cell is there, if it is undergoing meiosis, how many cells we will get? The product of meiosis. So you just think and answer. You just recall from meiosis, how many cells you will be getting. Or if you are watching live, you can comment that answer also. See, it is my microspore mother cell is there. Microspore mother cell. This cell is undergoing meiotic division. So how many cells it will be formed? You just let me know that how many are. So here microspore mother cell, when it undergo division, you will get 4 cells there. So that you will get 4 cells. This cell will form 4 cells. Of course, after meiosis of microspore mother cell, we will be getting four daughter cells there. These daughter cells, what will be the ploidy of each cell there? Ploidy in the sense a set of chromosomes present in that. Ploidy means, see, ploidy means set. How many set of chromosomes will be there in this microspores? See, these are daughter cells already. These daughter cells got ready already. So this is microspore mother cell. Microspore mother cell undergoing meiotic division. I get 4 microspores there, or 4 daughter cells, that we are calling it as microspores. So these microspores, so they will be wet in conditions like this, means which are having some moisture, some wetness will be there in this one. So these are what? These are microspores. So these are microspores, and the ploidy of each cell. What is the nucleus? The number of chromosomes in each cell there. The number of chromosome in each cell. What is the ploidy there? So the number of chromosome in each cell, it will be haploid in content. Here microspore mother cell is diploid in content. So when it is showing meiotic division microspores, it will be in haploid content. So through meiotic division. So now you can understand this definition here now. The process of formation of microspores from microspore mother cell. The process of formation of microspores from microspore mother cell through meiotic division. The process of formation of microspores from microspore mother cell through meiotic division. This process, we call this process as microsporogenesis. So we call this process as microsporogenesis. Now whether the pollen grains are ready now. So my question is, now microspores are prepared here. My question is whether the pollen grains are ready now. So you just think whether the pollen grains are ready or not. So still the pollen grains are not ready. Means it is the earlier stage of pollen grains. See, all these 4 cells are there, no? All these 4 cells, they are capable of becoming a pollen there. Each pollen grain it is going to be. So this one is one pollen grain, this is another pollen grain, this is another pollen grain, this is another pollen grain. It is going to become after maturation. So these microspores after maturation, means after losing the water or moisture, after losing the moisture, this covering is there. No, it is actually covered with some callus there. There is a material called callus which is holding all the pollen grains. Wetness will be there. No, that is due to this. So these microspores after losing the moisture, so it will form a pollen grain there. Means these are free from that moisture now. So these pollen grains are ready now. This is one pollen grain, this is another pollen grain, this is another pollen grain, this is another. So during the time of bursting of the anther, at the maturation of anther, it will burst. So when it bursts, the pollen grain will be dispersed like this. So it is going to shed the pollen grains like this in the form of this one. This is the dried pollen grains. So now whether it is a complete development of pollen grain, not yet. This is not the complete pollen grain development. So it is the baby of pollen grain. It is the again means earlier stage of pollen grain there. again this pollen grain has to complete so that we can call it as a complete male gametophyte. So there is another process is completely it will be taking place in each pollen grain. So that process we call it as gametogenesis, complete gamete formation of this one. So it is a pollen grain is, so you need to note here is the pollen grain. So pollen grain is male gametophyte. It is a male gametophyte. Pollen grain is a male gametophyte it is. So pollen grain is male gametophyte. So initially sporogenous cells were there. The cells will be the sporogenous cells will be acting as microspore mother cell. So after meiotic division, the microspore mother cell will become microspores. Microspores will become pollen grains. Those pollen grains after maturation they will distribute. But before maturation there is process still it will be continuing, no? So that we will be studying in pollen grain now. So that we will be calling as male gametophyte development. Here male gametophyte means, see, understand these lines. So here male gametophyte is there, no? Here male gametophyte in the sense they are talking about pollen grain. So the pollen grain development it will be in the last slide what we have seen, the formation of microspores we have seen. So this process we have seen, this whole process starting from here to here is there, no? This process we have seen, that is we call this as microsporogenesis. So that is microsporogenesis. That process is there, no? This process from here to here we are calling it as microsporogenesis. So here male gametophyte development, that is pollen grain is there, no? How the further development takes place here? See how the further development of pollen grain will takes place, that we will be seeing here. So the pollen grain is there. Say now initially what was there? Microspore mother cell was there. So this microspore mother cell it will form what? 4 microspores or microspore tetrad we call this as. So microspore tetrad it is. Microspore tetrad which is having 4 microspores, and from this each microspore, this is microspore. Microspore which is in microspore is in haploid condition. This microspore when it formation of pollen grains will be taking place, no? Pollen grain it will be in initially, the pollen grain it will be initially single cell will be there like this. The single cell will be there. It is pollen grain. Now this pollen grain which is having one nucleus of haploid content. So haploid content is there. So at that time they will show, so they will show mitotic division now. So it will show mitotic division. Mitotic division in the sense what? Mitotic division in the sense what? You just comment down, if you are watching live you just comment down what is mitosis there. So what are the there is another name for mitotic division also, you can comment down that also. Mitotic division in the sense what? Mitotic division in the sense the number of chromosomes it will be conserved, means n is there, n, 2n is there, 2n, like that. So the number of chromosomes n is there here, there will be mitotic division will takes place in this pollen grain. So when this pollen grain, so there is a mitotic division will takes place. So when the mitotic division takes place it will form 2 cells. So it will form 2 cells like this. means two cells or two nucleus it will form. It will form two nucleus. This mitotic division is asymmetric. This mitotic division is, so coming to this mitotic division, it is asymmetric mitotic division. You need to note here, it is very important. Asymmetric mitotic division. Asymmetric in the sense what? Asymmetric in the sense, you can see there is a big cell is there here, there is a bigger cell, there is a bigger cell is there, and there is a small cell is there. So it is a complete injustice, no? Means division taking one big cell and another one is small cell. So it is a big cell and it is small cell. This is asymmetric mitotic division, we call this as. And this stage of male gametophyte development is there, no? This pollen grain, we call this as two-celled stage. We call this pollen grain as a two-celled stage. Two cells are there within the pollen grain, two-celled stage of pollen grains. So some pollen grains in some plants, in some flowers, they will shed, they will release from the anther at a two-celled stage. And some pollen grains in some plants, in some flowers, they will shed at a three-celled stage. Three-celled stage in the sense what? See, the same one, it will be showing again another mitotic division. So another mitotic division will be taking place in the lower part, this one. So it will be showing another mitotic division there. It shows another mitotic division, the lower cell. When it shows another mitotic division, so now it will become three-celled stage. So initially one was there and it will become three now like this. So it is three-celled stage. Now it becomes three-celled stage. So initially two-celled stage was there, now it becomes three-celled stage. So another mitotic division here. So they may ask you question like to form a complete male gametophyte or to complete pollen grain, how many divisions it will take place? In the options they will be giving. One meiotic division, two mitotic division, two mitotic division, one meiotic division, three mitotic division, two meiotic division and one mitotic division. So like this options will be given. There how many are taking place to complete formation of pollen grain, how many divisions and what kind of divisions are it is taking place? So one is meiotic division here, one meiotic division, two mitotic division. So one meiotic division, two mitotic division, I am getting complete pollen grain, which is matured pollen grain, where it can shed, where it can release the male gametes. These are the male gametes. This is a male gametophyte. Pollen grain is a male gametophyte. Remember this one here. So pollen grain is male gametophyte, not a male gamete there. So it is carrying the gametes within that. So now what we have discussed is just a development from microspore, how the pollen grain is formed. It is just a development what I am discussing, and what we have seen is development of male gametophyte we have seen. So one is asymmetric division, we are getting two-cell stage, one is bigger cell and the one is smaller cell. And another one what we are seeing is another mitotic division, it will take place in the lower cell. That is, we call it as generative cell. This is vegetative cell, that we will discuss in the structure of pollen grain. That's why I am not explaining that. What we are focusing in this slide is only the development of male gametophyte. So how the male gametophyte will develop? So what are the divisions it takes place? And how are the divisions are? So two-cell stage and three-cell stage it will be taking place. Some angiosperms we said, no? Some angiosperms I said. So the some angiosperms means here, so 60% of the angiosperms, they will release it in the two-cell stage. and remaining 40% they will be released in 3-celled stage here. So remaining 40% of the cells, 40% of angiosperms, the remaining 40% of the angiosperms, they release it in 3-celled stage. They release it in the sense what? So if it is a plant is there, so it will be having pollen grains here. So if it is releasing, shedding out, it is releasing in the air, then it varies. Some 60% of the angiosperms, they will release the pollen grains when they are in 2-cell stage. So remaining 40% of the cells, they will be releasing the pollen grains when they are in 3-cell stage. So it is about the male gametophyte development. Next we are going to see the pollen grain now. Complete structure of pollen grain we will be seeing now. So now I'm drawing a layer here. See, pollen grain of course is a cell. You know that it is just a cell. So cell in the sense what will be there? A cytoplasm will be there in that cell, in the sense a cytoplasm will be there. A membrane is covering to that cytoplasm will be there, that we call it as plasma membrane. So here I am drawing a plasma membrane now. So it is cell membrane or plasma membrane. So it is plasma membrane. Outside to this plasma membrane, this is plasma membrane, and inside of this what you are seeing, this is complete cytoplasm, and this is plasma membrane. This plasma membrane, outside of this plasma membrane you will be seeing another layer there, another layer you will be seeing. So let me duplicate this one. Another layer is seen outside of this one. So another layer is seen outside of this one. So this is plasma membrane, and this is another layer which is covering that one, that is cell wall. That is, we call this as intine. The cell wall of this pollen grain, we call this as intine. And next to that what you are seeing, another layer you will be observed in this. So the another layer will be present, not like continuous like this, it is complete continuous surrounding this, no. The another layer which you will be seeing, it is irregular, not continuous. It is irregular and it is not continuous, and it is in many pollen grains of many plants, the irregular discontinuous structure that it will be having different texture in different plants. So this different texture and different it is there, no, even the chemical composition also differs. So we know that the innermost layer is plasma membrane. It is plasma membrane, and next to that what you are seeing, this cell wall, that is we are calling this cell wall in pollen grains as intine. So this intine composes of what? Cellulose and pectin. So it is composed of cellulose and pectin, the intine, and next to that intine what you are seeing, the irregular structure, that we call this as exine. That is exine. Exine is the outermost layer, and it is made up of a chemical called sporopollenin. So the special thing about here is, it is very important here, they asked 2-3 times on this. So it is important, the sporopollenin here. So exine of the pollen grain. So here exine of the pollen grain, it is made up of a chemical called sporopollenin, and that sporopollenin is the hardest organic substance which cannot be dissolved with any alkali or any enzyme or any acid also. So that is a hardest organic substance which is covering this pollen grain. Okay, so understand this. Sporopollenin has its own feature there. So it can be dissolved in, it cannot be dissolved in any organic acid, alkali, or any enzyme also. So for now we don't have any enzyme, acid, or alkali which can dissolve this sporopollenin layer. So it is important there. And due to the presence of sporopollenin, even pollen grains can be stored, like even it can be preserved in the form of fossil also. So this, because of the sporopollenin, it is protecting the pollen grain, no? So it will be protecting the gametes there. So this sporopollenin is having that much importance. So it is what? The sporopollenin is, the thing about sporopollenin is, it is the hardest organic substance which cannot be dissolved by enzyme, alkali, or acid. Or any acid. So that is about the sporopollenin. So that's why, because of that, even it can be preserved in the form of fossil. Preserved as fossil there. Because of that, we can preserve it as fossil. So it is about what? It is about the sporopollenin here. Sporopollenin is important here. And next to that, now what we have seen, it is of only external layers. Even in your book, you can check there in NCERT book, there are some diagrams are given. In the right hand side page, 3 diagrams are given above that. The electron microscope study of different pollen grain structures exhibiting different structures. Different textures has been exhibited by pollen grains that you can see in electron microscope. There it will be showing different textures over there. So here hexine is made up of sporopollenin and it is having irregular. So that those features also important. What I am discussing, that is also important there. See how is the intine here. The lines which I am using, that is also important here. Those are the NCERT lines. See what is intine? Intine is continuous layer which is made up of cellulose and pectin. If you see hexine, it is irregular, discontinuous. See somewhere there is absence there. There is no sporopollenin is present. So it is a discontinuous layer. Discontinuous and irregular in shape and different texture it will be showing. And there are some space where sporopollenin is absent. See here, you can see there is no sporopollenin. Here sporopollenin is not there. So why do you think that the place which is present even in pollen grains where sporopollenin is absent? Why? What might be the reason why the plant is leaving some spaces where sporopollenin is absent? So you just think why there is no sporopollenin here, and here. Just think why. What might be the reason? Why sporopollenin is absent? We know that sporopollenin is this one. This is the hardest organic substance which cannot be dissolved by anything. So then it will be helpful in protection, we know that. Then why these spaces, somewhere the spaces are left? Why? What might be the reason? So that is due to, and the space where the sporopollenin is absent, absent is there, no, that we call this space, this is a spore or a germ pore, we call this as, it is a germ pore. Germ pore in the sense it is kept where sporopollenin is absent. Here sporopollenin is absent, which will be kept open for pollination. So pollen tube formation. So it is opened for pollen tube formation there. Germ pore is kept open for pollen tube formation. Pollen tube formation in the sense, see, it is open here, it will germinate a pollen tube like this, through where the male gametes can pass. So the male gametes will be there, no, in pollen grains. To go through that, it germinates, it makes a pollen tube there. Through that the gametes will be passing there. Okay, so that is kept for that. The germ pore is for sporopollenin, where it will be absent there, and the pollen tube formation it will be present. Because of pollen tube formation it will be having that kind of structure. And if you see the pollen grain, inside the pollen grain, so inside the pollen grain we have discussed here in this last slide, it was only initially only one cell will be there, one nucleus it will be there, and one cell stays, that is pollen grain. So when the mitotic division takes place, asymmetric division takes place, when the asymmetric division takes place, it makes two cell stays. So when asymmetric division takes place, it makes two cell stays there. You can see there, this is a bigger cell here, it is a bigger cell there, it is the bigger cell there, and the lower one is a smaller cell. The bigger cell is there, no, which is bigger in size. That cell, the bigger size cell is there, no, that cell I am calling this as a vegetative cell. And the smaller cell is there, no, this smaller cell, this cell I am calling this as a generative cell. So there are two types of cells are there, what you are seeing here. So that is, so one, the cell which is bigger in size, the cell which is bigger in size, this cell I am calling this cell as vegetative cell. After one mitotic division, in pollen grain, it takes place, one is vegetative cell which is bigger, and another one is generative cell. This is generative cell. So vegetative cell in the sense what? So the vegetative cell in the sense which is bigger in shape, bigger in size. So I am writing the feature of vegetative cell now here. It is bigger in size. And why it is bigger? Because of asymmetric division it has taken place in pollen grain. Asymmetric means what? Which is not symmetric. So if it is cell is there, if it is dividing exactly center, it is symmetric. If I am dividing this cell like this, it is asymmetric, no? So the asymmetric mitotic division, it results in vegetative cell which is bigger cell. And a bigger cell has a large space and more cytoplasm will be there. So more cytoplasm or dense protoplasm or more cytoplasm it will be. So more cytoplasm means more food will be there. Means food reserve material will be there. Food reservoir. And the nucleus will be big here. So this one is asymmetric and this one is spindle shaped cell. So the thing is, when we see this cell as a asymmetric division, this is a vegetative cell, and in that this generative cell will be floating in this vegetative cell. So we know that vegetative cell is bigger. The generative cell which is having spindle shaped nucleus here, it will be having, see, this one is vegetative cell, and the another one is generative cell. It is generative cell which is having spindle shaped nucleus. It is generative cell. So generative cell is there. Generative cell is small. It is what? It is small in size. It is small. It is smaller. And spindle shaped nucleus. It is spindle shaped nucleus. Later, this generative cell only, after showing second mitotic division, so it is going to divide, it will form male gametes. So the generative cell only, it will become male gametes here. So the male gametes are formed here. So these are the feature of generative cell. And the generative cell, it will be floating in the vegetative cell, and it is having asymmetric division. Later another mitotic division will takes place. It forms two male gametes. So that is sperms. Two male gametes are formed. So two male gametes, it will be followed after the germination of pollen tube. It takes place. No, at that time it will follow. So this one, see, listen here carefully. Vegetative cell, it will germinate the pollen tube. So when it germinates the pollen tube, so this generative cell is there. No, this generative cell, it is going to divide into two male gametes. So these two male gametes will follow that pollen tube where the tunnel will be there. It will follow and it will fuse with the female gamete there. So this is it is about the structure of pollen grain. So far what we have learnt is, so one is microsporogenesis and male gamete development and the structure of pollen grain. So the today topic, we have completed almost a page of information. You can check there in your NCERT. And related to this, we'll start next session by asking the question only. So we'll start the next session by solving some questions which are related to these topics which we have discussed today. So I hope you understood everything whatever I thought, and you enjoyed the session with this colorful diagrams and information which we have discussed. So true NCERT is there, complete NCERT information you can check and you can read. If anything is there, if you are facing any difficulty understanding this lecture, you can comment down so that we can I can answer those questions in the next session. Okay, thank you.